We'll get started. So Taylor is kind of a unique situation with energy. He got involved a little later. We had started marketing this to the KCU and Campbell folks, but Taylor came to us late last semester, right, Taylor?
Yeah, that's correct.
Yeah. Okay. But he's been great so far. Really showed initiative and an ability to be a self-starter, and clearly is on the path towards success. I think he's interested in neurosurgery, but we will definitely help him to get there. So he's a second year at KYCOM, which is in Pikeville. I don't know where that is. You said Eastern Kentucky?
Yeah, Eastern Kentucky.
Okay. He graduated from Toledo and University of Toledo in 2018 with a Bachelor of Science in Biology and a concentration in neuroscience. While he was there, he participated in research labs dedicated to studying optogenetics and molecular signaling pathways that influence neuronal behavior. And I'm not going to even try to say the name of that organism. But since entering medical school, he's co-authored several articles. I think he and I are actually co-authored on one. And his most recent one is, I know, one that was published in Curious on BCI and Neuralink. He's the class president for class of 2023 at KYCOM and enjoys exercising and following the news of his favorite football team, the Steelers, which I cannot believe that Ben Roethlisberger is coming back. But anyway, I will let you take it away, my man.
Awesome. Thanks, Mike. And yeah, I can't believe he's coming back either. I was not too happy to hear that. Let me go ahead and share my screen here. Okay. Okay, can everyone see my screen okay? Okay,
You've got you got the weird presentation mode.
Oh, I have the weird presentation mode. Okay.
If you go to display settings, you should be able to change.
Let's try that.
Yeah, there you go.
Is that better? Okay, perfect. And then can you also see my cursor as well? This was an issue yesterday. You can see my cursor. Okay, awesome. Thank you. Okay, well, like Mike said, my name is Taylor Reardon. I'm a second-year student at KYCOM, and I want to thank you all for having me here today. The title of the article that I'm sharing today is Hemoglobin Induces Oxidative Stress and Mitochondrial Dysfunction in Oligodendrocyte Progenitor Cells. I think this is a really interesting article that has a lot of cool and important implications in neurosurgery in the future. But the title is a little bit wordy, so we need to talk about some background info. So there's a common pathology that comes up in very low birth weight infants called intraventricular hemorrhage, or neonatal intraventricular hemorrhage. And it's essentially the infiltration of blood into the cerebrospinal fluid and surrounding brain parenchyma. And this occurs in up to 30% of those very low birth weight infants. So it's a very common pathology that's seen. The mechanism for how this occurs is that there are vessels in the germinal matrix, which is right next to those big lateral ventricles inside the brain. And in premature infants, just like everything in premature infants, nothing is usually fully developed. And so these vessels are they can rupture very easily and cause that hemorrhage into those ventricles. And inside the blood, there are products such as hemoglobin and iron that you know of. And these products can build up inside those ventricles and lead to further pathologies such as post-hemorrhagic hydrocephalus and matter injury. And the white matter injury can also lead to other pathologies later on down the road, such as cerebral palsy and seizures and mental deficiencies. And this is just an example of some imaging that you would see in an infant with intraventricular hemorrhage. The first one on the left is a coronal view, and as you can see on the right, which would actually be the patient's left, there is this white blob in the middle, and that is signifying a intraventricular hemorrhage. And then comparing the middle and the far right, the middle is actually the intraventricular hemorrhage in a sagittal view, and then the right is a normal view. So you can see that there is less space inside those lateral ventricles. So when a baby is developing, there are these cells called oligodendrocyte progenitor cells, or OPCs as they're commonly referred to. And these are the precursors to the oligodendrocytes. They give rise to white matter in the CNS and develop the axons. And a trait that they have is that they're highly vulnerable to oxidative stresses and reactive oxygen species. And not only are these cells susceptible, but the mitochondria, the powerhouse of the cell, is very susceptible to ROSs and the damage. And the problem with damage to mitochondria is that if you damage the mitochondria, you can't produce a lot of energy for developing cells to develop. And this can cause the cells to either stop developing altogether or severely inhibit their ability to develop. So there's a drug called phenelzine, or PLZ as it's referred to in this presentation. It's a FDA-approved antidepressant or anxiolytic that has a monoamine oxidase inhibiting mechanism, but it has been shown in previous experiments to have strong antioxidative effects to restore the mitochondrial functions in the animal models of brain injury. And the mechanism for that is that it binds carbonyl groups via hydrazine group, and scavenges and eliminates the free radical species in the brain. So knowing all of this background, the hypotheses that were presented in this paper, they were divided into two hypotheses. The first one is that they believe that hemoglobin can induce oxidative stress and mitochondrial dysfunction in these cells. When you apply hemoglobin and blood essentially to these cells, that there is going to be a decrease in functioning of these cells. The second part of this is that this drug phenelzine can be used and applied to these cells to reduce that oxidative stress, restore the mitochondrial function, and increase proliferation of these oligodendrocyte progenitor cells. And this is a really nice schematic that the paper included that kind of separates their hypotheses into two different sides. The first hypothesis is actually on the left, where when you apply the hemoglobin to these OPCs, you're going to see an increase in reactive oxygen species with subsequent decrease in respiration, ATP production, and ultimately decrease in the proliferation of these cells. And then the suggested mechanism for the OPCs when you apply phenelzine is that it will suppress those reactive oxygen species, increase the functioning of the mitochondria, and subsequent enhanced OPC proliferation. So it's basically supposed to restore the effects of hemoglobin on these cells. So the ways that the investigators investigated this is they isolated these cells from Sprague-Dawley rat pups postnatal day 2. And to measure the reactive oxygen species, they used something called a DCFDA fluorescent dye. And the mechanism behind this dye and why it works is that this dye is applied to these cells and it only lights up, it only shows immunofluorescence when there's oxidation. And so if there's no oxidation present, these cells won't light up. So that will confirm the presence of some oxidative process going on. The second part is observing how the mitochondria works. And this is measured in oxygen consumption using what's called a seahorse assay. And I'll get into how that works in a second here. And then finally, cell proliferation. And this was analyzed using a BRDU incorporation assay, which essentially looks at how BRDU is incorporated into the proliferating cells and using immunofluorescence as well. So the cells that are proliferating properly are going to light up, and the ones that aren't are going to stay stained with the identifying stain. So let's get into the first round of results here. In figure 1A at the top left, you can see that the cells were stained with NG2 and DAPI. And then the third image there is just merged together. This just confirms the presence of the oligodendrocyte progenitor cells. The way that the cells were isolated was that they took the whole brains out of the rats. And so there are a lot of cells in the brain, as you know, and they want to make sure that they have OPCs, and they harvested with accuracy over 90% in the populations that they had. And in figure 1B, this was a measurement of intracellular reactive oxygen species, or ROS generation. The white bar shows the vehicle, and then using amounts of 0.05 milligrams and 0.1 milligrams of hemoglobin, you can see that there were statistically significant increases in ROS generation in those cells. And anytime you see an asterisk above a bar, that is indicative of statistical significance. And for this paper, they used a p-value of less than 0.05. In figure 1C down below, this is the application of that DCFDA assay and immunofluorescence dye. And as you can see in this vehicle, it's very blue. I hope you can see, it might be a little hard to see, but it's very blue in the first pane, indicating that there's not really a lot of oxidation going on, but there is still a basal level of it. And then as you apply hemoglobin, you can see that there is lighting up of the cells, showing that oxidative process and the immunofluorescence from that DCFDA. And this was quantified in figure 1D, showing that it was the y-axis indicates normalized intensity of the immunofluorescence. And this shows that application of 0.1 milligrams of hemoglobin does cause oxidation, which is visualized by that DCFDA assay. And then in figure 1E, they split the experiments into 3 hours and 24 hours. And the reason why the authors of the paper did this was that the pathology of intraventricular hemorrhages, it's not just an acute process. It's not here's a brain bleed and then here are the effects and then they're gone as soon as you remove the blood, or they're gone as soon as you do whatever you need to do. The effects of the brain bleed can stay for days or even weeks after it occurs. So they wanted to investigate once you take away from, or I'm sorry, they want to see the long-term effects of hemoglobin on these cells. So they measured the reactive oxygen species in these cells when they were exposed to hemoglobin for both 3 hours and 24-hour intervals. And you can see that there is a significant generation of those ROSs at both of those time points. So that first figure there proved essentially part of the first hypothesis of the paper, in that hemoglobin does cause reactive oxygen species generation in these cells. Now, the second figure is going to try to address the application of phenelzine to these cells. And so if you look at figure 2A here on the left, the vehicle down below has that base level of reactive oxygen species generation. And then you apply the hemoglobin, the same amount as in the previous experiment, 0.1 milligrams, and there's a significant increase in ROSs. Now, the main comparison I'd like to draw is between bars 2 and 4 here. And bar 4 shows that there is a group of cells that were exposed to hemoglobin with 10 micromolar of phenelzine. And with a p-value of 0.053, just missed statistical significance. There is an observed trend, though, that it does decrease reactive oxygen species inside the cell. But what was interesting to see is that as they increased the concentration of phenelzine from 10 micromolar to 30 micromolar to even 100 micromolar, there's actually an increase in the amount of ROS generation inside these cells, suggesting almost ROS protective effects of these cells. Now, there's no statistical significance between those measurements, but it can still be observed. And then looking at the figure 2B, there was even a lower correlation between phenelzine and reduction of ROSs inside these cells. So it was interesting to see that it didn't cause really any effect in the 24-hour group. And it was also interesting to see that the base level of ROSs was higher in those cells. Moving on, this is the first half of the results from the seahorse assay. And at the top is what's called a seahorse cell mito stress assay. And there's a lot of dots and points here, but essentially they divided the cells into 4 groups. They had vehicle groups of 3 hours and 24 hours and hemoglobin exposed groups of 3 hours and 24 hours. And what they first did was measure the baseline oxygen consumption rate of the cells to get what are the cells doing at the base. Second, they administered oligomyosin to the cells, and oligomyosin is an inhibitor of one of the components of the electron transport chain. So you're going to see a drop in the oxygen consumption rates because it is affecting how the mitochondria work. This molecule here called FCCP is a uncoupling agent. And what this essentially does is turn the mitochondria into overdrive. They need to work at maximal respiration in order to maintain the functions, or maintain the normal functions that they would if they didn't have the oligomyosin or any other stresses to it. So this FCCP is used to observe the maximal respiration of the mitochondria in each of those cell populations. And then finally, the ROT anti-A stands for rotenone and antimyosin A, which are further inhibitors of the electron transport chain, which basically renders the mitochondria almost not able to produce any energy. And this was used to measure the non-mitochondrial oxygen consumption. And so if you go through figures 3B through E, you can see an interesting pattern in that there was no change in the basal respiration, the ATP production, the maximal respiration, or the non-mitochondrial oxygen consumption in the 3-hour groups, showing that, suggesting that maybe hemoglobin doesn't have that strong of a effect on the mitochondria in an acute setting. However, when you look at the 24-hour group, there's a significant decrease in oxygen consumption in basal respiration, ATP production, and maximal respiration. And there's no significant decrease in non-mitochondrial OCR, but there is an observed decrease. So moving on with the next experiment, the authors of the paper decided to not even use the 3-hour group because there was no change observed in those cells when exposed to hemoglobin. So this data is all from the 24-hour cell groups. So in the first experiment, we have our vehicle, which is the 24-hour groups, and you can see that the oxygen consumption rate is near 100%, meaning that the mitochondria are working the way they should. When you expose hemoglobin to those cells, it drastically drops. And then when I want to draw another comparison here, which the authors did between bars 2 and 4, this shows the application of phenelzine to these cells. And although it's not statistically significant, it did show a p-value of 0.07 and observed a trend in the increase in restoration of basal respiration. And they tried it with 30 micromolars and 100 micromolars, and there was no observed or no statistically significant restoration, in any of those. But they did find that there was a statistically significant restoration of maximal respiration when using 10 micromolar of phenelzine in the 24-hour group. So there was one experiment out of the 4 that did show some promise in restoring function in the mitochondria. And then in the bottom there, there's non-mitochondrial oxygen consumption rates, and you can see that there's almost 0 change across the board in the 24-hour group as well. And then finally, here is the BRDU incorporation assay, which was used again to observe the effect of hemoglobin and phenelzine on the proliferation of these oligodendrocyte progenitor cells. And so in pane A, up in the top left, this was just the vehicle. And the BRDU, I'm not sure if you can see it, the BRDU is the red. So if you look at the pane there, there's presence of red, there's presence of the identifying immunofluorescence stain, the DA. And then when you go over to pane B, with the application of hemoglobin, you can visually see that there is a decrease in that red stain, the proliferation of those cells. One of the hypotheses that they said was that phenelzine alone, not even in the presence of PLZ, would enhance the proliferation of these cells. And so that was measured in pane C. Just adding phenelzine, would they see an increase in proliferation? And looking at it, you can see that there is a little more red in comparison to pane A. And then finally in D, that is the phenelzine and hemoglobin group. And drawing the major comparison between panes B and D, you can see that phenelzine did somewhat restore that red tone or the red hue that you can see. But unfortunately, going over to figure E over here, you can see that there was no statistical significance across the board, actually, with the parameters they used. There was not even a statistical significance in the percent proliferation when they just added hemoglobin. So the authors did state in the paper that they were kind of disappointed by these results and surprised as well with all the effects that they had found in the previous experiments, showing that it did affect mitochondrial function. It did produce reactive oxygen species, so they thought that maybe it would cause issues with proliferation. But based on these experiments, it did not show statistical significance of that. So I'd just like to kind of go over, it was a lot of data, a lot of results, but I'd just like to kind of go over what we saw and then wrap it up with some thoughts. So the hypotheses, they hypothesized that hemoglobin significantly increased intracellular oxidative stress in these cells, which it did. The first graphs did show that there was an increase, in ROSs in these cells when you applied hemoglobin, both at 3-hour intervals and at 24-hour intervals. What was not expected was that when they went to the mitochondria, the effects of the mitochondria were only observed after those 24-hour groups, and there was no change at the 3, which supports the notion and the pathology behind intraventricular hemorrhages, and that you can see the effects of this in the brain and in these OPCs after many days in the clinic and in these cells. And then phenelzine reduced the oxidative stress of hemoglobin. It was shown that it did have an effect on that. It did reduce mitochondrial respiration and ATP production. There was only one of the experiments, as we talked about, that did significantly decrease, but there were suggestive trends. And then finally, unfortunately, there was no significant effect of either hemoglobin or phenelzine on OPC proliferation, which was surprising, but could be further investigated in subsequent experiments. So the big question is why does this matter? Why is this an important paper, I feel like? Well, the role of phenelzine is not the only drug that has been investigated as an antioxidative therapy in IBH. They've investigated erythropoietin and vitamin E, and we could see a increase in the FDA approval of these drugs in adjunct therapy in intraventricular hemorrhage in the near future. To treat these, they usually only have to treat the symptoms, and it usually involves either a spinal tap, inserting a shunt, blood transfusions if it's due to ischemia, things of that nature, but they can really only treat the symptoms, and there's not a whole lot they can do with the subsequent neurologic sequelae in the developing child. And so these experiments almost keep progressing in the promise of implementing almost a neuroprotective agent that could be applied with these surgical techniques in order to protect the baby a little bit better from neurologic damage as they develop if they are born prematurely with an intraventricular hemorrhage. So I thought that this was a pretty exciting paper to share, and looking forward to further developments in this field. So I just want to thank you all again for letting me come by. It was great to present, and there's my contact information down below. If you ever want to email me or anything or have any questions, just let me know. And they're my sources.
Taylor, good job. Did you have any, did you come across anything in, because this is all translational work, have they looked at this in humans at all yet?
Specifically not phenelzine. They have not looked at phenelzine yet. I believe that there was a study that I read where they were looking to do something with erythropoietin, but I believe it was a clinical trial, but I don't know the results.
Okay. And did they talk about, why they chose phenelzine specifically in terms of its antioxidative effects?
Yeah, so it was found in previous literature that there were experiments done using phenelzine that reduced reactive oxygen species productions in the brain in general. They did investigate phenelzine. I don't know the reason why it was used before then, but there was an experiment that did show that it did reduce those in the brain in general in animal models. And so this group kind of took that and ran with it and applied it to specifically oligodendrocyte progenitor cells.
And then my last question is, did they look at other primary glial cells in terms of, do you know of other studies that have looked at other glial cells?
I know that the same group,
astrocytes or ependymal cells, or.
Yeah, exactly. I know that this same group was looking into working with microglia. And I believe they would, I can't remember, I actually contacted the author of the paper and asked this exact question, and they were looking into doing the experiments with microglia. They had plans to use astrocytes, but with COVID, it kind of put a wrench into plans with that. So I would expect this group to probably work with microglia in the future, and other groups as well, I would assume.
Great, thanks.
Dr. Wolf, I see your hands up. Go ahead, please. Yeah, I actually have several questions. You did a nice job of presenting this. I guess among the first questions that I had was how long was the hemoglobin exposure? Was it continuous or was it just a brief exposure?
It was a brief exposure in the experiment, but I don't know exactly how long it was for. I know it was brief. I know it wasn't like a continuous exposure from the isolation of the cells to like the 3-hour or the 24-hour mark. I just don't know how long it was for. Sorry.
Well, I guess the reason I'm asking the question is when babies have this hemorrhage, I mean, that iron is going to be generating reactive oxygen species as long as there's iron that's not been oxidized. And so with that being the case, it seems to me that one would be interested in getting the hemorrhage blood out of the ventricle as quickly as possible. Do you know if that happens?
I do know that it's, I mean, that is the goal, is to get it out of there, but sometimes because of the amount of blood that is produced, it's not necessarily detected because sometimes there's no neurologic sequelae that's necessarily detected in the babies that have the intraventricular hemorrhage. And that's why sometimes the physicians do notice that there might have been, the blood could have been absorbed, or you might not be able to notice it on an ultrasound, but there is detection of reactive oxygen species damage in these babies days or weeks after they have one.
Well, and so I guess I was also going to ask before I forget magnesium. So they give magnesium to preemies, potentially even the mother, with the evidence that that protects against, it doesn't necessarily delay labor and delivery, but there's clear evidence that it protects against neurological damage. So have you factored some of that into your thinking here?
I know that is used. I didn't think about it in these experiments, honestly, but that would be interesting to see if.
Well, I didn't look it up, but it seems to me that isn't superoxide dismutase magnesium-based thing. So to that extent, you might be expanding on the antioxidant activity, which I guess leads me to my final thing, and that is, it seems to me that when you have iron as an exogenous cause of reactive oxygen species, you are consuming endogenous antioxidant capability. And among the major sources of endogenous antioxidants is the electron transport, or of endogenous reactive oxygen species is the electron transport chain. And so it seems to me that it makes complete sense to me that their data would be obtained because they consume the cell's ability to sop up reactive oxygen species while the mitochondria would continue generating them in the absence of appropriate antioxidant pool. So I'm not surprised by their data at all, but then again, I'm just wildly speculating too.
Right. Yeah. I thought the same thing reading through it. You're not that, like you said, it's the, most endogenous producer of ROS species. You knock it out, it's going to produce a ton. So that, like you said, the data does make sense. It's just interesting for me to see mostly the proliferation of cells. I was very surprised to see that wasn't affected. And also too, the amount of their N, and this is, a common, gripe about experiments, your N wasn't big enough, but really the N for these experiments, especially in the proliferation assay, I believe the N for this one was 4 brains. So would we see a trend that would show statistical significance if it was 40 or 400? Maybe, but I don't know, unfortunately. It would be cool to see though.
Well, I guess the other silly thing that crossed my mind was, do the cells heal? I mean, because the mitochondria are damaged, but they should be undergoing fusion and fission. And through that process, they should be able to hopefully generate ones that truly work.
Mm-hmm.
So thanks. I'll shut up now.
Thank you, Dr. Wolf. I appreciate the questions.
Yeah, thank you, Dr. Wolf. I had a feeling that you would have some thoughts regarding some pathways when it came to some of these drugs and what's happening with the ROS there. So thank you for that. And thank you again, Taylor, for your presentation and for, energy for bringing us another Journal Club series. We do appreciate it. It's Friday, so I will say have a nice weekend, everybody. I know we do have some more talks upcoming. You guys are all on the invite list, so join if you can. And, if you want to come back and do another one, just reach out.
Taylor, I actually have one more question. It's more.
Yeah.
Sorry, Eddie.
It's boards. It's actually boards related. So, so I know you're studying for step 1, but it's kind of step 2 type material. So if a baby is having, if you're concerned, at what week prematurity should you be worried about intraventricular hemorrhage? And if you don't know, just guess.
Yeah.
You do your best guess.
I would say probably around 24-ish, I would say would be a very worrisome time.
So you're totally right. I mean, when you get that premature, you're obviously most concerned about lung maturity.
Mm-hmm.
So it's actually 30 weeks.
Oh, 30,
and
okay.
So at any baby born under 30 weeks should get what within the first 3 days of life?
Oh gosh. I'm not sure, honestly.
For intraventricular hemorrhage. Like what do you, don't overthink it.
Is it just, I don't know, is it just TPA?
So you said it. You said that you don't know if they have it, right? Because you might not.
Oh, imaging wise, like an ultrasound. Okay.
Exactly. Yeah. So you got to get an ultrasound within the first 3 days to screen for it in babies under 30 weeks.
Okay.
So just to the point of, whether or not you can do some kind of adjunct phenelzine with, draining it. There is an impetus to do that because you're looking for it anyway, right? You're not just treating it based on symptoms. So Anyway, just thought we'd bring some clinical correlation instead of talking about seahorses and whatever else you were talking about.
Are you giving them vitamin K then too, right off the bat?
So that's also something that is very common, and that actually, fits for multiple things. But yeah, that's a good point. Eddie, did you have a question?
No, I didn't have a question. I was just going to tell Taylor, very good job with the presentation.
Thanks, Eddie.
All right, now I'll shut up. You guys have a good weekend.